Hello everyone, it's high time for grade 11. These days we are talking about digestive system. This is lesson part 4. So we have discussed what is digestion and the parts of digestive systems or organs of digestive system and about the physical digestion. Today's lesson is about chemical digestion. So we will start our lesson with the digestion of carbohydrates. You can see in these pictures there are four sites where the chemical digestion of carbohydrates occur: mouth, stomach, duodenum and small intestine. Let's see how this process takes place. Actually, what are the carbohydrates that we take as food? One, starch and sucrose, also glucose and fructose. But glucose and fructose, they are already small molecules. Starch and sucrose, slightly large molecules. Starch is a polysaccharide and sucrose is a disaccharide. So before absorption, we have to break them into simple molecules. This breaking process we can't do physically. I mean cutting by teeth or churning in the stomach, we can't do that. So chemical support or enzyme support, we need to break these glycosidic bonds. So the enzyme secretion starts at mouth. Actually secreted by the salivary glands. The enzyme is called as salivary amylase. The salivary amylase is not enough to break whole starch amount, but salivary amylase start breaking of a starch. Some part of starch will break into maltose molecules, but some part remain. So there's no special enzyme comes from the stomach for carbohydrate digestion. But the food without changing pass into the duodenum. At duodenum, another amylase enzyme comes from pancreas. Therefore, we call that enzyme as pancreatic amylase. Pancreatic amylase will break the rest of the starch into maltose. So by that process, maltose will produce. These maltose molecules also little bit large and they have glycosidic bonds. We can't absorb that. We need to break them into glucose, the simplest sugar. Therefore, there is another enzyme secretes by the small intestine walls. That enzyme is the maltase enzyme. Maltase enzyme break these bonds, these glycosidic bonds. Then final product is glucose. So by three steps, the starch will break down into glucose. End product is glucose. Now let's see how does sucrose break down. There is no enzyme secrete by mouth or at mouth or the stomach or duodenum. No enzyme secrete to break down the sucrose bonds. Therefore, sucrose comes unchanged to this place, that is the small intestine. There's enzyme secrete from that. That enzyme is the sucrase enzyme. It breaks sucrose into its basic molecules called as glucose and fructose. So this is the carbohydrate digestion process. Starch and sucrose finally all break down into their monomers called glucose and fructose. Let's move on to the next digestion process, chemical digestion. Digestion of proteins. How does protein digestion take place? Look at this protein molecule. Protein molecules are very long chains of amino acids. We have to break them into that simple molecule monomers called amino acids. Look at this. There's no particular enzyme secrete on proteins at mouth. Therefore, it comes unchanged to this place. At stomach, there is an enzyme secretes on protein. It's called pepsin. Pepsin comes with the gastric juice. Pepsin need acidic media. That acidic media also provided by the hydrochloric acid comes with the gastric juice. Somehow this protein now will break down into slightly long chains, but they are shorter than normal protein molecules. So these polypeptides should be further broken down. Therefore, another enzyme secreted by trypsin at duodenum. This trypsin will break these polypeptides into peptides. Peptides are not the simplest or the shortest ones. So these peptides also should be broken down further. Therefore, peptides will come to the small intestine, and from the small intestine walls, peptidase enzyme secretes on peptides. Finally, amino acid units will be separated like this. The end product is the amino acid. Then proteins also broken down by three steps with three enzymes: pepsin, trypsin and peptidase. End product is amino acid. The special fact, that is renin enzyme. It is secreted only in infants, those who are breastfeeding, and those ones have special enzyme. It converts the water soluble proteins to water insoluble state, because that proteins in the milk, the breast milk, should be converted before the normal digestion. After it makes into the water insoluble state, then by the normal process of breaking down, we will break them down to the amino acids. This renin enzyme in infants will be secreted until they feed by the mother's breast milk. Once it is stopped, renin enzyme also slowly or the gradually stops. Then let's see the digestion of lipids. Lipid digestion. On lipids, special enzyme does not secrete by the mouth. Lipids normally we take as the lipid layers, or lipid exist as that. So this lipid first will travel to the through the mouth. Nothing happens because no enzyme secrete on that. When it comes to the stomach, there is no enzyme secreted by the stomach as well. So it comes unchanged to the duodenum position. In duodenum, there are two special liquid secretes at lipid. One produced by the liver and stores in this gallbladder, finally secrete. It is called bile. Remember, bile is not an enzyme, but bile can break these lipids into small fat or the lipid globules. It increases the surface area. Then enzymes can more effectively act on lipid globules. This process is called as emulsification. That means converting lipids into lipid globules. Then the enzyme comes. The enzyme secreted by the pancreas, it's called lipase. This lipase breaks this lipid molecule into its basic components. Lipid molecules are made of fatty acid and glycerol. So this is how it breaks down. Fatty acid and glycerol molecules, three glycerol molecules separate. So the end product is fatty acid and glycerol, which we can absorb into the body. So let's move on to the other part is this one. This small intestine doesn't secrete any enzyme for the further digestion. And the special thing here, I highlighted that one before as well. It is bile. It is not an enzyme. It produces at liver and stores at gallbladder. So remember that fact as well. Then let's move on to the last part here. Functions of gastric juice. And I said that with gastric juice, sometimes with gastric juice always an enzyme secrete, as well as hydrochloric acid comes with the gastric juice. Gastric juice helps the chemical digestion as well as it support the proper activation of the enzymes. These are the functions of gastric juice. Number one, destroys bacteria in our food. And next one, promotes the protein digestion by providing optimum pH value for trypsin enzyme, and provides optimum pH value needed for enzyme. That's what I said just before. Last one, decomposes calcium carbonate. Sometimes we take calcium carbonate when we eat small fishes. Sometimes we eat with the bones. Those small bones can be decomposed by the hydrochloric acid in the gastric juice, and those are the functions of gastric juice. Finally, with that one, we will end the part 4 of the digestion lesson. Next, we will meet back to learn about absorption process and the diseases associated with the digestive system.